Thank you everyone for attending Science Matters Seminar Series. Today, we are pleased to welcome Dr. Ghayda Mirzaa, Associate Professor of Pediatrics at the University of Washington School of Medicine in Seattle. Dr. Mirzaa received a medical degree from the Arabian Gulf University in the Kingdom of Bahrain. She performed her residency and clinical genetics fellowship at the University of Chicago. She then joined the University of Washington and Seattle Children's Hospital in 2012, where she started her research lab. Dr. Mirzaa received several honors and awards from her work in studying human malformations and morphogenesis, as well as neurobiology of disease in children. She's one of the expert panelists for the Clinical Genome Resource, or ClinGen, which is one of the key tools we use in rare disease studies. One of Ghayda's main research goals is to understand disorders affecting early brain development to find ways to treat and cure them. Today, she will provide an overview of how genomics has advanced our understanding of epilepsy disorders and how this knowledge informs clinical care and target therapies. Thank you, Dr. Ghayda, for sharing your work with us today.
Thank you so much. It's really a pleasure to be here. I really want to thank you for the invitation. It's really a pleasure to be here and to talk to you about some of our work. And I look forward to our future collaborations and discussions. So my work has for now many years has focused on the genetics of a subset of epilepsy disorders in children that are more surgically, amenable. So a lot of these kids undergo early brain surgery, and I'm really delighted to talk to you about how we started off with genomics, and now we've really kind of migrated towards more targeted therapies based on the genomic or genetic findings. So it's been a truly rewarding journey along the way. As an outline, I'm going to touch base upon three key themes here. I hope they're of interest to the audience. The first one is really from starting from my home base, which is genetics, and really how did we come about to understanding some of the underlying genes and pathways that are associated with these conditions. A lot of this was through deep sequencing technologies. The second is really about some of the findings of like brain-specific, genetic abnormalities, including mosaicism. What are the diagnostic applications for that? And then I'll highlight some examples of really fascinating and complex genotype-phenotype correlations, which I think are really interesting and challenging problems that we need to work on moving forward. And then lastly, the future of where we feel like this field is headed, which are more along the lines of minimally invasive molecular diagnostic tools and molecularly guided therapies. So these will be the three key themes of my talk. I hope you find them of interest. Just as a very general starting point here, what is the impact of the problem we're trying to study? So epilepsy overall, of course, affects, as we all know, a significant fraction of people. An estimated 65 million people worldwide live with epilepsy, 3.4 million of which live in the United States, more or less. Certainly, as anybody who's ever known a person with epilepsy or had a family member with epilepsy would know, epilepsy oftentimes does not exist in isolation. When it usually occurs, these individuals usually have a lot of other, what are called comorbidities that can include cognitive deficits, irrespective of the epilepsy, behavioral issues such as ADHD, autism, sleep disorders, psychiatric issues such as depression and anxiety, feeding dysregulation. So there's a lot of other things that go along with it that oftentimes in the clinical setting, we might not do a great job at really understanding the impact on the individual. These are really important from a genetic perspective as well, especially when we talk about clinical trials and targeted therapies. For a lot of families, besides epilepsy, these are really important potential targets that need to be addressed. In spite of that, there's other layers of challenges. The diagnosis and medical management are complex. And we know now very well that early diagnosis is really crucial for appropriate and effective treatment. And the treatment modalities are multiple options, so we have anti-seizure medications, we have dietary options such as the ketogenic diet, surgical approaches, which I'll talk about a lot because we've worked a lot on these conditions, And last, but certainly not least, are molecularly guided therapies such as mTOR inhibitors and other small molecules and drug targets that affect the pathway. Now, I have studied the brain for most of my career. I find brain disorders really fascinating. And what specifically I think we've been working on have been structural brain abnormalities that occur in children. So oftentimes these are congenital. Children are born with these abnormalities, more or less. And they span a really broad spectrum of phenotypes with different types of structural aberrations. So over here, and I'm sorry, the resolution is really not great. You have like what's called a smooth brain. Typically the cortical gyral pattern is highly folded. This is what's called lissencephaly, or a smooth brain. Over here, actually, you'll see disorders of brain size. So this is a small brain, so that's microcephaly. I don't know if my mouse is showing. And here you have macrocephaly, which is a big head on this side. That is actually one of the ones that I'll focus a lot about. Or a large brain size, basically. Now, the classic teaching about these brain malformations is that they were originally classified according to the stage of neuronal development shown here in the middle, during which the abnormality might have arisen. So, if people think about these as disorders of neuronal proliferation, migration, and organization. And that actually framework was really helpful for a very long time. That's the classic teaching. What happened was with next generation sequencing largely, so a lot of the genetic technologies, when they came about, we started to understand the pathways that are related to all of these brain malformations. So now the picture looks like something like this. So we've got all the pathways that we started to understand. And here's my pathway that I'm going to talk about up here, which is the mTOR pathway, which is a cell growth pathway. But so we started to kind of think about things from a different framework, which is a pathway perspective, similar to what's been done in oncology, right? And what's really helpful about this framework is, again, it puts us in a better spot to think about then molecularly targeting these pathways, as has been done in cancer for decades, right? So we can follow in the footprints of a lot of really amazing cancer research. So to hone in now on the specific group of disorders that we've been working on, these are called brain overgrowth disorders, or megalencephaly, which means a big brain. This is a broad spectrum, actually. So these are brain MRIs here that I'm showing. And what I would like to show you is that these kind of range from diffusely enlarged brains. So the entirety of a child's brain here is abnormal. Sometimes it's without any cellular dysplasia. So the structure is normal, but it's just big. Other times, like in this case here, it's actually extremely abnormal and dysplastic. You see lots of irregularities and abnormalities in the structure of the brain. Then we have more focal forms of overgrowth. So it could be half of the brain, so half of a hemisphere, what's called hemimegalencephaly. You could have a quarter of a brain, which is called quadrantic dysplasia. So that's shown over here. And then last, but certainly not least, you can have a very small area of the brain that is abnormal and dysplastic, and it's aptly called focal cortical dysplasia. So that means it's a focal area where the brain is abnormal and dysplastic. In the classic literature, if you look at it, these were previously, decades ago, clinically described by, mostly clinicians, and they were called different things. So they were believed to be different disorders, and these are the terms that were used. We now know through the molecular discoveries that they're all linked by mutations in the same pathway. So instead of thinking of these as distinct entities, we now think of them as a spectrum of involvement. So I'm going to pick on focal cortical dysplasia for a quick second, because it's really A, the most common, and B, it's really severe and has significant effects on affected individuals. So FCD, again, it kind of looks trivial on a brain MRI scan, but actually it really causes some of the most severe types of epilepsy we see. And many of these children, if their seizures are not effectively managed, suffer early death, basically. FCD has been neuropathologically classified, just like with a lot of cancers, for example, and for any of you who has studied tuberous sclerosis, for example, it has been neuropathologically classified into 3 broad subtypes, which has been really helpful. Type 1, which means there is isolated areas of cortical dysplasia, and you see like there's even like almost like molecular staging, 1A, 1B, 1C. Type 2, which is associated with very distinctive cellular abnormalities that I'll talk about in a second. Type 3, which means cortical dysplasia adjacent to other important lesions, such as brain tumors, vascular lesions, and others. Now, focal cortical dysplasia type 2 is actually really interesting because it's really now we know is the most genetic subtype. Most of the genes we find are in FCD type 2. Types 1 and 3, we still don't understand well at all, at least from a genetic standpoint. We've been doing a lot of genetic research. We don't find mutations in those children. In FCD type 2, we find a lot of genetic mutations. So the cellular characteristics are really interesting. There are two key features that are seen in the brains of people with FCD type 2. The first is actually what's called the balloon cell, which is shown on the first row here. This is where the cell is very enlarged. It's usually associated with an extremely ballooned out cytoplasm, and hence the name balloon cell. These balloon cells oftentimes stain positively. For those of you who do immunostaining, they stain positively for phospho-S6, which is an mTOR pathway marker. The second cellular feature is what's called hypertrophic neurons. So these are neurons by lineage, but they're large and dysplastic looking, and they're hypertrophic. So these are the two key cellular features of FCD type 2. And you'll see here, it says 2A have dysmorphic neurons, but without balloon cells. Type 2B have dysmorphic neurons with balloon cells. So balloon cells are a very sensitive finding, if you will, in children with type 2B. And so, again, this is now the most genetic subtype associated with this. So to segue into the genetic findings, more than 10 years ago, when I was doing my training, I did my medical school overseas at the Arabian Gulf University. And then I did a summer work. I was interested in neurogenetics. So I went to the University of Chicago through a mentor of mine who helped me out and got me into a summer internship at the University of Chicago. And at the time, I wanted a project, basically. So I was like just shopping around and begging, really. Through lots of fortuitous kind of collaboration, Dr. Ian Glass, who I don't know if any of you have known him, he's one of our longest standing dysmorphologists at Seattle Children's Hospital. He's been here for like 30 years, basically, I think. There was like a dusty pile of clinical records on like 5 or 6 kids with a very interesting combination of features, but it didn't have a name, this entity, you know? So these kids had a big brain, as you can see here, like there's a prominent forehead with an enlarged brain here. The ventricles were big. Actually, there's sometimes some of these kids have hydrocephalus or fluid in the brain. There's something called polymicrogyria, which means that the surface of the brain, you can see the red arrows. The surface of the brain, which is supposed to look like a walnut, it looks very abnormal and dysplastic. So a lot of the time it's associated with seizures. And then something called polydactyly, which means extra fingers. So this is an X-ray that shows actually a 6th finger. So we called this syndrome, not very creatively, perhaps on my part, MPPH syndrome, after the 4 key features associated with it, you know, megalencephaly, polydactyly, extra fingers, PMG, and hydrocephalus. We published this in 2004, and we thought that was kind of the end of that story. Fast forward a few years, next generation sequencing was coming on the scene. The UW was spearheading and really revolutionizing the field by next generation sequencing. You know, the Genome Sciences Center was really at the cutting edge of this. And so we prioritized samples from kids with this syndrome for exome sequencing. And lo and behold, we found mutations in 3 genes in this pathway: PIK3R2, AKT3, and cyclin D2. So all 3 genes in MPPH syndrome associated with it. And they're all within the same pathway and at different nodes within the pathway. And I won't spend more time talking about MPPH syndrome, except to tell you that since then we've continued to study this condition. We have more than 100 families worldwide with it. We're doing genotype-phenotype studies. There's actually a possible link between a subset of these kids and brain tumors, specifically medulloblastoma, and specifically with kids who have cyclin D2 related MPPH syndrome. And there's a whole lot of interesting research and conversation that can be had about cyclin D2 and its role in the development of medulloblastoma. So we're trying to explore this link further to see, it seems to be a genuinely increased risk for medulloblastoma in these children. So we basically kind of tried to ride this wave, right, of genetic advances where, you know, historically people prior to NGS, we all had like more limited options, including looking at chromosomes, single gene testing, and then we moved on to multi-gene testing, exomes, and now, of course, genomes. We've got both short and long read genomes around. If you look at, this is one of my favorite graphs, which is actually quite old now, but I'm aging myself, but like if you think about like this is just for epilepsy genes, you know, prior to NGS, we had like what's called the channelopathy era, which was more standard sequencing. The rate of gene discovery was kind of slow. And then we had like a little bit of a black spot here, like, and then next generation sequencing came and bam, you know, like gene discovery just kind of shot up. And it has continued since actually quite a bit. So what does that mean for our brain overgrowth disorders? Well, most of the genetic discoveries have pretty much converged on the mTOR pathway. Exomes, pretty much, and most NGS to date has converged on this pathway in these brain disorders that I talked about. And the mutations really range in this pathway. So it's not the same class of variants. So we have gain of function mutations of activators of the pathway and loss of function mutations of inhibitors of the pathway. So you kind of have the same functional endpoint, basically. You're either like inactivating a brake or pressing on the gas pedal. And essentially you get activation of the pathway as a whole, which is a growth pathway, right? So this isn't entirely surprising, but the key point that I want to make is that it's not just one gene and one mechanism. So you've got heterogeneity here. So this led us at the University of Washington. I work with a phenomenal team of scientists and molecular lab directors who we decided to design a panel then to test children in the clinical setting. So we designed a panel for this called the Megaplex panel, which does deep sequencing for these genes efficiently and is available. So I'll show you an example of a child we tested. So this is a child who was followed at Seattle Children's for many years. She was born with a very abnormal and dysplastic brain. You can see here, one half of her brain was abnormal and dysplastic. And because of that, she had very early onset epilepsy. And so essentially surgery was really life-saving for her. So she had a lot of that hemisphere surgically removed. She also had, and I'm not sure if this is showing that well, she had a little vascular lesion here. And you can see it here, this red patch on her leg. So what we did at the time, we were able, because of the extent of her lesion, we had multiple brain regions to test. And we did a skin biopsy. So we found, through the Megaplex panel, we found an AKT3 mutation, which is the E17K variant, in multiple brain regions. These are the tissue locations, and in the skin lesion as well. What I want to call out is if you look at the brain, the levels of the mutations ranged from about 10 to 18% in different brain regions. And these are the actual raw data, like if you wanted to see like how many mutant reads and how many reference reads specifically. But what's really interesting is that if you look at the skin lesion, even though this is a vascular abnormal skin lesion, the level of the mutation was much lower. It was 1.3%. You could argue that if you do standard sequencing, this could be missed, right? We had 10 reads that were good quality, so we were confident, but if we had poorer coverage, this could easily be missed. So it's very important. It taught us a lesson that it's very important to have as good quality data as possible, of course, but more importantly, perhaps, is actually assessing the right tissue. And of course, brain, most of us, myself included, would like to hold on to as many brain cells as I can. So, that is an important consideration. So this led us then a few years ago, one of my research scientists at the time, Milena, in conjunction with our amazing team, our neurosurgical team, these are our 2 neurosurgeons at Seattle Children, and our neuropathologist extraordinaire, Dr. Raj, who's on this corner. Sorry, my mouse is all over the place. So this led us a few years ago to design a project where we took our repository. We have a repository of fresh frozen brain tissue samples and a few FFPE brain samples for a total of 124 brain samples from children that are collected clinically at the hospital. A lot of these families are generous and they are willing to donate additional samples from their children to research. So we had additional samples at a smaller number, like fibroblasts, very small numbers, but saliva, we had a good number, I think, blood and skin biopsy samples, for example. And we decided to ask a very specific question as it relates to mosaicism. We wanted to ask basically, how low can we go? So what is the lowest level of mosaicism that we can find in these brain lesions? And in order for us to ask that question, we had to make the inevitable compromise between breadth of sequencing and depth of sequencing, right? So when you do these studies, you kind of have to decide how much of your DNA do you want to assay, and compared to how deep you want to go at the base pair level, right? So we wanted to go deep, which means we had to compromise and we had to really be very targeted in our strategy. So we designed 6 probes for the 6 most common mutations in these genes that are also most commonly seen in cancer, by the way. So these are highly oncogenic mutations, highly activating, 3 in PIK3CA, 1 in AKT3, which is the same one I showed you earlier, 2 in mTOR. So just these 6 hotspots using droplet digital PCR, and we tested our repository. And this is a plot actually showing the mutations that we identified in different tissues, shown in a color-coded format here. And the x-axis is showing each individual who tested positive, and the y-axis is showing the level of the mutation. And what you'll notice is that for some of them, we had multiple brain regions that are shown in black. And so you can see that there's a range of mosaicism in brain tissue, which was actually helpful because a lot of the literature, you get data that is one tissue, one data point per tissue, which is great, but it's still not, it's helpful to see what is the range across the same, tissue, for example. So that helped us to kind of look at the level of mosaicism, the range of it in the brain in these children. So it can range actually from like 0%. Sorry, where's my mouse? My mouse is disappearing on me. Here we go. From 0% all the way up to like almost, close to 15%. What you'll notice is that all other non-brain tissues kind of hover near the bottom, specifically blood. So we're oftentimes not finding these mutations in blood. You will notice that saliva has a bit of a better yield, which is interesting. And that's a conversation to be had about why that is. So what are some of these disorders and some of these genes? So one of them is called PIK3CA. PIK3CA is actually a gene that's upstream in the mTOR pathway right here. And it's a very important gene developmentally, and it's associated with the whole spectrum, brain spectrum that I showed earlier. But actually, if you look more broadly, there's similarly a diverse spectrum of phenotypes that are seen in the body. So PIK3CA can be associated with severe body overgrowth. You can see here, look at this massive lipomatous overgrowth with vascular lesions, these vascular abnormalities with massive overgrowth of a limb, fingers, cheek, neck region. So the same picture of a spectrum of overgrowth in the brain and the body. If you look at the clinical literature, each of these was called a different name. These are just the acronyms, which are just completely your eyes glaze over when you see how many clinical terms were used, and they were believed to be different disorders. But now we know it's basically like the case of the blind individuals and the elephant, right? Which is very common across any scientific discipline, is that depending on which part of the elephant you may be studying, you think that explains the entirety of it. But in fact, you learn that it's a whole thing. And so we now know that all of these disorders, actually, a few years ago, a group of researchers got together and they said, we need to clean up, the whole classification. These are all linked by one gene. And molecularly, we need to rename this PIK3CA-related overgrowth syndrome, which is the broad term that is now used. So it's really helpful for understanding the spectrum. So from this study that we did, we wanted to look at a few clinical parameters. We weren't able to get a lot of detailed clinical data on a lot of the kids, but we definitely had some. So the first question we asked was, does the presence of one of these mutations impact or have a relationship with the onset of epilepsy? And what we found actually was a statistically significant finding, which is that kids with one of these 6 highly oncogenic, highly activating mutations, very bad mutations, actually did have earlier age of onset of epilepsy. Typically, it was around, a year more or less, whereas the negative ones was actually, over, much over that. So we did find a correlation. So that meant that the presence of one of these mutations is really associated with earlier onset epilepsy, which is really helpful. So now what I wanted to kind of mention is that these findings kind of challenged us because there were 3 hypotheses in the field at the time that were present, and I think we learned a lot about looking at these 3 hypotheses related to this pathway. The first is that these mutations are mostly mosaic. They are mutational hotspots, and they overlap with those causing tumors. So that's the first hypothesis that's been around. And I'll show you what we learned about each of these very quickly. The second hypothesis that's been out there is that these mutations, if they are present in a germline state, meaning in 100% of the body, they would be embryonically lethal. So these are not compatible with life, and that's why we see them in the mosaic state. I'll show you what we learned about that. And then the last one, actually, which is a lot dicier, is that the classic teaching in genetics, at least in the clinical practice, is that the levels of mosaicism correlate with severity, meaning the lower the level of the mutation is, in theory, these people should have milder phenotypes, whereas individuals who have a higher level of mosaicism usually have a more severe phenotype. I'll show you what we learned about that. So starting off with the first one, these gain-of-function mutations are overlapping with that we see in cancer, so in adults with cancers, not kids with overgrowth syndromes, but adults with different varieties of cancers. So what did we see related to that? So we started with PIK3CA, which is by far the most common gene. This is a diagram of the gene. On the lower half, I'm showing you a plot from the COSMIC database for cancer. If you look at that database, it's like a database of a wide variety of cancer tissues in humans. And it's showing you the different mutations. You'll see the 4 hotspots in PIK3CA. 4 or 5 more or less. And extrapolated on that, we added the mutations that we see in these brain overgrowth disorders and body overgrowth disorders. And what I hope you can see is that the hotspots are mirrored. The peaks are the same across both developmental disorders and cancer. So that hypothesis seemed to hold true. But what if we looked at some of the other genes, right? So we took a step back and we looked at, these are cosmic plots for PIK3R2, AKT3, and mTOR, where we find mutations. And indeed, the peaks in each of these genes in cancer, in adults with cancer typically, are the same ones we see in pediatric overgrowth disorders. So these hotspots are real. So essentially, for example, the G373R is the most common mutation in MPPH syndrome. The E17K variant in AKT3 is the most common variant in AKT3 related disorders. And similarly in mTOR, the S2215Y, which is in the kinase domain, is also the most commonly seen in pediatrics. So highly recurrent variants, right? So that was helpful. The second hypothesis was, so that hypothesis seemed to hold. Second hypothesis was that these mutations have to be mosaic in order for them to survive. And if they are germline, they are embryonic lethal. Well, it turns out, starting as, as far back as like 7 years ago, we started getting contacted by clinicians across the world who were saying, I have a child with a germline PIK3CA mutation. So that means it's present everywhere, present in blood. And we started collecting their data. We started collecting the mutation information and their phenotypic data. We now have a cohort of around close to 40 individuals with germline PIK3CA mutations who have a broad neurodevelopmental disorder. So they have head overgrowth as shown here. So they have dysmorphic facial features. You can see actually the big head a little bit with the prominent forehead here. They can have neurodevelopmental delays, they can have autism, they can have a whole range of other features as listed over here. And what we did is we looked again at the PIK3CA gene to see where does that relate to what I showed earlier, the hotspots that are seen in cancer. Where do those mutations fall? So here on the lower side, I'm showing the hotspots, so these are the most common ones. And what you can see now is that the germline variants, which are shown at the top, are different. So these are typically milder mutations in other functional domains, and they are present in the germline. And in one instance, actually, they were inherited from a parent who was affected as well. So that means that these mutations were compatible with longer life and reproduction and, and so on and so forth. So the second hypothesis proved to be now incorrect. The third one, which is that the levels of mosaicism correlate with the severity of the phenotype. The very simplistic way that, I thought about it for many years was that if you look at the mutation load, in theory, it should be that kids with, smaller, localization to the mutation, lower mutational load should have milder phenotypes. And it's fascinating when you see brain MRI scans that sort of look like this, right? So I showed you earlier, where it's like a small part of the brain. This could be FCD. This could be almost quadrantic dysplasia here, and this could be hemimeg. But in fact, it's a lot more, this turned out to be a much messier question, right? Because there is a mutation distribution, but there's also, but clinical severity is defined differently. Clinical severity is often defined by how bad are the seizures? How bad are the developmental delays? It's a whole different framework of thinking about severity. It's not just about looking at the scan and seeing how much of the brain is involved. It's really looking at the child and seeing what are they showing. So we tried to answer this. This is still a work in progress. We looked at 550 children with these phenotypes, and we tried to, and we had more than molecular data on more than 350. And we were looking at the ranges of mosaicism in the 3 most common genes in different tissues and across these 3 different phenotypes, right? So what we found is that the kids who have diffuse brain overgrowth more or less had germline variants. So that means they were present everywhere. And oftentimes, the reason we don't have brain values is because these kids have diffuse brain overgrowth. Most of the time, they're not going to get surgery, right? Because they've got symmetrical bilateral involvement. They're not going to be surgical candidates. Kids who have hemimegalencephaly oftentimes do get surgery. So we find mosaic mutations that are present in the brain and in the body, variable degrees. Whereas kids with the most restricted lesions, FCD, typically have very brain-specific mutations that are often absent in other tissues. So that was helpful to kind of try to parse out that question. But more interestingly, for some of the kids where we had multiple brain regions, we felt that maybe let's dive deeper into now the cellular landscape here and look at the severity at that single brain level. So this is an example from one child. He was in one of our website, like on our website, actually at Seattle Children's many years ago, in a very like lovely piece and to his family, because the child, the family donated the brain tissue to research, but he had extensive, involvement shown by the arrow here and underwent resection. You'll see here, it's actually post-surgical, the part of the brain that was removed. And the dots here are showing all the different regions from the brain where we had actually access to DNA. And we found an mTOR mutation in him, which is shown here, which is the common one. And then we decided then to look at the cellular pathology across these regions to see the level of the mutation ranged from 0%, even though the tissue was abnormal and surgically removed. Some regions were negative, to 9%. And what was interesting is that we saw neuropathological abnormalities across all of these regions. So there was a breakdown between the genotype-phenotype relationship in that sense. So some of the regions that were 0% still had the same cellular abnormalities, balloon cells, dysmorphic neurons that are seen in some of the cells that are mutant positive. So this hypothesis is a very open-ended question, but we all know that genotype-phenotype relationships are complex. And that I don't think is going to go away anytime soon. I think we'll continue. This is part of the new, the things we need to continue to study beyond just finding the mutations. So what this led us now to, this is the final kind of summary of that, is that we have like almost like a framework of like thinking about these disorders, that they are typically a spectrum depending on the level of mosaicism. And you can have, if you have low level that are mutations that are brain specific, typically you'll get focal cortical dysplasia. If you have intermediate level mutations that are present mostly in the brain, but a little bit in the body, you get, different involvement. And then germline variants, you get kids with a big head with neurodevelopmental issues and sometimes autism and other things. As a teaser, I'll also mention that there have been, most of the time, these are isolated instances in that family, but we've had several families where the family would have multiple children who are affected. So this is a family where mom and dad had 3 children with MPPH syndrome, and we found this PIK3R2 mutation. Now, our understanding was that these are de novo mutations. So it's a random mutation that happens. We would find it typically in one child. So the fact that it happened in 3 children means more likely that one of the parents has the mutation, whether at a low level or in their gonads only, for example. And we think it's gonadal mosaic because we tried to do deep sequencing of saliva, accessible samples, for the parents, and we couldn't find the mutations. Here's another family with the same gene, the same syndrome with 2 affected children. So parental gonadal mosaicism, we believe, occurs in these disorders. And actually, if you look at the literature, there are at least 3 case reports of gonadal mosaicism, whether maternal or paternal, with multiple affected siblings. These are some of these case reports in this pathway. So it's a fascinating spectrum. The 2 parts, these are kind of interlinked, so I've lumped them together. It's like the second half and last half of my talk. I wanted to give now more practical kind of lessons we've learned along the way. So the first one is really a word of kind of a word of caution that we've learned about. In that mosaicism really, I think, is an important consideration for a lot of this. So this is a girl who actually was born with a big head, and she actually had hydrocephalus. So there's actually a shunt here that's not very clearly shown. She's got a shunt track. She didn't have a lot of vascular lesions or anything, but she had a big head and she had hydrocephalus. And we felt like she had one of these, syndromes and most likely an mTOR pathway mutation. But we only had saliva and blood, samples. So we started by doing targeted testing. This was at the time using molecular inversion probes, which were revolutionized in Jason Dure's lab at the U. So we did that on saliva, deep sequencing, 400 to 500x at the time, negative. Exomes came along a few years later. We did exome sequencing on both blood and saliva, and we tried to get deeper coverage in saliva. So we did 200 to 250x, negative. Then whole genomes came around, right? So we did whole genome sequencing, preferably on blood. Saliva genomes are hard because you've got a lot of bacterial contamination and you're going to amplify a lot of non-human sequences. So you get a lot of, it really reduces your sequencing, basically the yield of your human data. So, and at the time, this was actually many years ago, even before the protocols have improved, also negative. So many, many years later, the family finally agreed to do a skin biopsy. And we did deep exome sequencing on that skin sample. And we found the cause, right? An mTOR mutation. And it was seen at less than 10% in the skin sample. And what is this really, what is the point, here? The key point that we learned at the time is that, we took the family, and I wanted to show this here. This took about 10 years. What's considered a diagnostic odyssey, right? And this is not a journey that you're just taking yourself. It's really, you're taking a family with you. So, and this was hard because this family was really sad, by the end of it. I mean, the child had passed away later on because of her severe neurological features. So it's quite sad. And in the sense, I think we kind of felt responsible in that we really took the family along this diagnostic odyssey when in fact, and we learned about mosaicism somewhere down, somewhere through here, right? But still, I think the key point is in order to test these kids, you really need to have you need to have the right tissue oftentimes. And if you don't have it, sometimes just going through all of this, it can be a little bit difficult. So it's a little bit of a lesson. So this led us actually a few years ago to say, well, can we optimize? Like sometimes we don't have the tissue. Can we optimize our analytical tools so that even if we have a peripheral sample, can we find these low-level mutations? So one of our very talented bioinformatics team at the time, led by lead bioinformatician Andrew Timms, asked a question is, could we actually design a pipeline to look at our data, exome data, from blood or whatever tissue you have, but to actually optimize the pipeline tool to look at mosaic variants. So we had like two types of pipelines that were longstanding, and many labs have this, a germline analysis pipeline for exomes. And then we have a copy number, a very standard copy number analysis, which with exomes, of course, is a little, tricky. But so then we came up with a mosaic pipeline. So we looked at some of the cancer callers, like the one we actually settled on the most was called Pisces, which really allows you to pick up low frequency variants. And we said, let's use this pipeline that is used in cancer to actually look at our existing exome data. We have exome data from 583 children, 638 samples total, and we reviewed the clinical data. And what was really helpful is that for, most of the time when you look at studies, exome studies, you look at what's called de novo variants, right? Which is a child, you compare the child to the parents and you find something that's unique to the child. And we certainly did that, and it's very important to do that. But what we did actually was a parallel analysis as well, because we have usually paired samples from these kids. So we actually did what's called a somatic, pipeline, where we actually compared then the child's brain sample to the germline sample. And I'm not going to go through all the results from this exome study because it's a lot of things we found. But when we looked at like the different phenotypic groups, and this is just a very busy table showing all the genetic findings from this exome study, what I want to call out is that when we looked at how many we solved by exomes, you'll see like across, so this is microcephaly or a small brain. This is other malformations of cortical development. You'll see kind of like an even split between solving, finding something, finding a candidate, which is shown in blue, and unsolved, which is shown in yellow or orange, I don't know. But what's really interesting is that one group stands out where you have a whopping unsolved situation, and that's our brain overgrowth kids. So these are the kids with the spectrum that I showed you. And even in spite of these analytical tools, we still don't. And the reason, of course, to us at least, was mosaicism, the fact that we don't have brain tissue on a lot of these kids. So this led us to really understand now that we've got three key things here to look at, which is you have your sequencing sensitivity. So the more you optimize the sensitivity of your method at the single cell level and tissue level, the better. The balancing factor to that is your genetic heterogeneity. Again, you often have to make this very challenging balance, whether from, for scientific reasons and also from a sadly financial reason as well, in terms of the cost of these techniques. You often have to make the compromise between deep sequencing versus broad sequencing, right? So that's kind of a very challenging balance because we have many genes in this pathway. And actually, we actually believe that there are other pathways that are involved that are understudied. So we've got genetic heterogeneity. So how much can we broadly and deeply sequence? The last one is obviously very self-explanatory, but is probably the deal breaker for most of the time, is tissue availability. So we're privileged at Seattle Children's to have an amazing neurosurgical team who's been extremely supportive of genetic research. I want to show you an example of a child, actually, with a fascinating phenomena that is still unexplained to us. And I'd love to hear thoughts about it, maybe offline if like, the, just for time. But this is a child who had bilateral brain involvement. And because of the bilaterality of the brain involvement, the child unfortunately had very early onset seizures and was not a surgical candidate. So he passed away at around 10 weeks of life. He was seizing pretty much from birth, so, and unfortunately passed away. And family donated the brain to research. The child's brain weighed 905 grams. The expected weight at that age was 506. So you can see how big these brains can be. We didn't have fresh frozen tissue, so we had paraffin embedded blocks for the most part, and this was all formalin fixed very early on. This was an out-of-state situation. So we had a really hard time because we didn't have a lot of DNA, and we were very concerned about using the very little and valuable DNA we had. So we sat on the samples for a while, but then once we had our panel coming along, and our panel is used in oncology and things like that, so we can use it on scrolls of tissue and paraffin embedded blocks and everything. We found first an AKT3 variant, which is shown at the top. This variant here at 38%. We were like, all right, this makes sense. This is a gene we know is, implicated. We thought we had it. Then what Milena did actually when she designed the droplet digital PCR experiment, she threw, you need smaller amounts of DNA for ddPCR. So we threw in the DNA from this child in our ddPCR experiment. Lo and behold, we found 2 additional variants, different ones in PIK3CA at a very low level, less than 1%. Both of these passed multiple technical replicates because they're low level. We were kind of worried about a artifact, but we did our best with like quadruple checking everything. We usually have a panel of controls, positive and negative controls, and they panned out every time. So essentially what we think happened here is 3 mutations. And the question is, can 3 mutations occur, right, in the same brain? And could that explain the really severe phenotype? This is among the most severe cases we've had across the years. So that's an interesting question. It's still out there, so I'd love to hear if anybody has any thoughts. And then I wanted to show you another challenging case where also we still don't really understand it fully, but it's along the lines of genotype-phenotype correlation. This is a child who had a resection of the temporal lobe, and we had a superior temporal lesion and an inferior temporal lesion for DNA. And we found, and sorry if the font is small, we have found a very low level PIK3CA mutation, 0.24%. So again, less than 1% of cells in the superior temporal, if I recall, in one of them, and then 0% in the other region. So our amazing neuropathologist asked the question, well, let's take a look at the brain slides from both of these respective regions to see if there's a difference between the region that's 0.2% and the region that's 0%. So he did laser capture microdissection. So we had 2 regions from the superior temporal, 4 regions from the inferior temporal. And the mutation was 0 in the superior temporal, both lesions. So we did drop the digital PCR on each of these. And it was 0.1% and 0.2% in 2 of the 4 inferior temporal. And I'm not going to go through all of this because this is a massive amount of work. Through microdissection of each of these regions, what we found is that, as a summary, really, but I'm happy to go through this if anybody is interested later on, we found that in both of these regions, we had both dysplastic regions that had dysplastic neurons, dysmorphic neurons, and balloon cells, and non-dysplastic. So you'll see here dysplastic and non-dysplastic microdissection. And similarly here, in both the regions that are completely negative for the mutation and regions that are positive. And again, this suggested that there is a breakdown between the genotype and the phenotype in these regions. And it raises a whole set of other hypotheses about then how does the phenotype occur? Is it actually due to the mutation itself? Is it the mutant cells that actually harbor that are associated with the phenotype that are dysplastic? Or is it actually cell signaling, right? Is it cell signaling? Is there non-autonomous cellular effects here at play? Lots of interesting challenges. So this is the last part. I think, am I doing okay on time or am I over time? We have 15 minutes, right? So this is the last part, I promise. This is now leading us to the next wave of, and I'll show you what we mean by that. So what do we know so far? Germline samples are challenging for these because the diagnostic yield is low. We don't often find mutations. Brain samples, sometimes they're inaccessible, unavailable, or you don't get enough DNA, right? So this raises another interesting avenue that people have been looking at, which is cell-free DNA, which is now a new thing. And specifically for these disorders, so these are fragments of DNA that are free floating, right, in various fluids in the body, plasma. This is now used prenatally for diagnosing, chromosomal abnormalities and other things during pregnancy. This is considered now standard clinical care to do it from maternal cells without doing invasive fetal testing. The question is, could we do it in CSF, right? Could we actually do that? So, and there's a lot of literature coming out about this actually for brain tumors, right? That they can actually find cell-free DNA in CSF from individuals with brain tumors and do actually molecular testing on CSF rather than brain tissue testing, which is a lot more invasive. And what's really interesting is that actually the main mechanism is that it's neuronal cell death. These cells die and they release the DNA. Aberrant neuronal death has also been observed in epilepsy patients, not just brain tumor patients, and in animal models. And I'll show you, this is a case, I'm not going to go through the whole clinical story because it's very long, and this child had a lot of complications, but he's actually now an adult. He lives in Oregon. He had huge brain overgrowth and vascular lesions. He basically fit the clinical picture. He had a lot of other comorbidities. He had intestinal lymphangiectasias. He had lots of GI issues secondary to that, significant developmental delays and behavioral issues. The brain was bilaterally involved, so we never had brain tissue. He never required epilepsy surgery or anything. He did have hydrocephalus, and he had a laser ablation just cosmetically for one of the vascular lesions. Well, lo and behold, he got really sick at some point right after his 18th birthday. He had recurrent pleural effusions, and he had atypical lymphocytes in the pleural fluid, and an excisional biopsy showed that he had a B-cell lymphoma. And so what happened is that he underwent a lumbar puncture, so a spinal tap for tumor staging. And we decided then to take an extra sample of CSF and look for cell-free DNA in that sample to see if we find the mutation in him. And what I wanted to show you here are those results. So he had a PIK3CA mutation, which is not surprising given the phenotype. What was interesting is that we found the mutation in his CSF, in the cell-free DNA, at a low level, 3%. This is a very small amount of DNA, relatively speaking, for us at least. We hope to usually get a little bit more. And these are actually the plots from the mutation, what are called IGV plots, to show the level of the mutation in the 3 tissues we eventually had on him. The cell-free DNA from CSF, skin, and blood. And you'll see actually in the skin, it was 37%. In the CSF, it was around 3%. And in the blood, it was around 2%. So this showed us that we can actually potentially find these mutations in CSF it's still invasive, obviously, but less invasive than brain testing. So we published this as a report not too long ago. I'm going to skip this one, actually. And there's actually been several papers now that showed that cerebrospinal fluid liquid, what's called a liquid biopsy, these are all called liquid biopsy-based methods, are actually really practical potentially, and they can actually help detect these mutations. This is a proof of concept study where they found these mutations in LIS1, TSC1, and BRAF. BRAF, of course, is well known in the brain, and then they found it in the CSF as well. So wrapping all this up, we know that we have, a very important subset of disorders with an extremely important impact on these children. And we do have therapeutic endpoints that I'll talk about in a second. But we've got lots of challenges here, right? Lots of parameters we got to, keep in mind in terms of tissue distribution / accessibility, genetic heterogeneity, cell autonomous and non-autonomous effects, and so on and so forth. And what is the therapeutic promise here, right? mTOR, for any of you who've studied the mTOR pathway, probably that this has been heavily targeted by pharma for many decades. There's a wide range of mTOR inhibitors on the market, including dual inhibitors. You can see here some of the dual drugs that target multiple targets, and then single inhibitors. The most famous one, obviously, is rapamycin, or everolimus, which has been used in oncology and used for tuberous sclerosis. There is this one also, which is a newer one called alpelisib, which actually came around a few years ago because it is used, FDA approved, to treat PIK3CA positive breast cancer in women. It has been used in kids with PIK3CA vascular lesions because obviously there's a clear clinical trial design there, which is you can give the drug and actually see if the lesion shrinks. So it makes it an easy therapeutic endpoint because you could do imaging and see volumetrically how much these lesions go down. So it was actually found to shrink these lesions. So it had beneficial effects in kids with body overgrowth from not just the overgrowth perspective, but even if they had scoliosis or back curvature problems, vascular lesions, quality of life, pain, and mobility, which makes sense. The big question that was out in the field is CNS effects. We don't know. actually, what it does in the brain, more or less. So now where we are is that we have now, so, you know, in the omics era, right, the era we are in is tissue-specific genomics. And we really need to do deep sequencing on brain tissue. So we have a clinic at Seattle Children's where we see these patients. We offer testing, whether it's the megaplex or deep exomes / genomes is another question. And I try to then offer post-test counseling and see if they can be enrolled in a clinical trial using one of these drugs. And our testing paradigm is similar to oncology, basically. It's paired tissue testing. However, this is my last one, I think, hopefully. My last slide. Are we missing stuff, you know, genetically speaking? We absolutely are, because so far, all the data that I showed you on mosaic mutations in the mTOR pathway pretty much explain about 50% to 60% of kids. So we do know we are missing other mechanisms. Some of the hypotheses include copy number abnormalities, structural variants, as we talked about earlier. Non-coding variants are always a possibility. Methylation abnormalities are actually another possibility. This was actually an interesting study that's not too long. There's been multiple studies, and this one is actually not too long ago, where it actually showed that structural variants involving AKT3, where you can have extra copies of AKT3 that are mosaic also, which is a challenge. So these could be mostly brain specific, are associated with these phenotypes. Oh, this might be my last slide. I'm sorry. Like this is actually now there's a new modality also now called stereotactic EEGs in children with epilepsy, where they put implant multiple probes in the brain, do recording from multiple regions to see exactly to localize where the epilepsy is originating from. So it is invasive, but it's actually utilizing probes. So it's minimally invasive. So that way the whole field is moving towards extremely minimally invasive resective surgery. You know, we want to preserve as much brain tissue as possible for these kids. So this is actually a neat study where they did also a proof of concept, and they took they washed off the cells that are attached to the probes from these SEEG probes, tried to take these cells and purify them and extract DNA, and they were able to find mosaic mutations from these probes at different levels, which means we could potentially avoid the kids having to have brain surgery if we can. If they're eligible for that. And so they found these mutations using droplet digital PCR actually in some of these kids from these probes. So they did targeted testing and they found mutations from, you know, you can see here 3 probes, E1, E2, E3 with their corresponding EEG. So you can actually have the genetic data in parallel to the EEG data, in parallel to the imaging data, which is usually high resolution. And you can compare all 3 together at the same time, so which is great. So this is my wrap up, I think. I'm sorry if it went a little over. I was hoping to have more time for questions. I hope this was of interest. These studies, I can't emphasize more. I was the mouthpiece, to be honest, for a lot of work here, because these studies do take a village of amazingly talented and passionate people about this work. So I want to really acknowledge our neurology team at Seattle Children's, our neurosurgeons, our neuropathologists, our lab medicine team, our medical genetics team, the Genome Sciences Center, and my lab, as well as our funding sources, and of course, as always, the families who have supported the research over the years. And thank you so much. And I'm happy to take any questions.
So much, Ghayda. I think this is a genetic class that you give us. So thank you so much. Really, really nice and interesting, a lot of work. Any questions?
I saw one, looks like Lisa.
Oh, should I?
So
Lisa has her hand raised. So you could invite her to unmute.
can invite her to.
I don't see. Sorry, I don't.
Yeah, so
She can.
Oh, she can. Okay. Oh, I don't need to.
am I unmuted? Good. Okay. I just wanted to, first of all, just fascinating talk and just mind-blowing in terms of trying to think about diagnosis. I have a million questions for you. I get to talk to you this afternoon, so I'll ask most of them then. But let me just ask one, which is, if you have a low percentage of mosaicism in a particular tissue, I'm thinking of the brain in particular, but it also could be other cell types. Often, if a mutation arises, it depends on when it arises during development, right? Whether it's going to be in a whole lineage or just a small lineage. And I just wonder if there's anything on the horizon in terms of looking at single cell populations, for example, to see if there's it just happens to be the GABAergic neurons here or something like that.
Yeah, absolutely. There's been some single cell work that's been actually done at Boston Children's led by Chris Walsh and his team, where they've actually taken whole brain tissue, and have been looking at like neuronal versus glial, cell lineage, and then GABAergic versus glutamatergic, which would be extremely important and helpful. So yes, there is some of that a little bit. We still don't have a clear consensus on which cells are typically mutant in these phenotypes. I mean, balloon cells, the lineage of them actually is still somewhat debated because they stain positively in different studies for both neuronal and glial markers. So they're somewhat confusing in that sense. And these are hypothetically believed to be the most, the mutant, the ones that would harbor mutations. And this has been contested a little bit by the TSC literature, which actually has showed that some of the, in TSC, you've got these lesions called cortical tubers. And what some of the single cell work that's been led by people like Peter Crino and others has shown is that when they did microdissection and to actually try to get as many single, or tissue localization as possible, they found that mutations, actually the tubers were often mutation negative and the perituberal tissue was mutation positive. So it kind of, was confusing, and we still don't know, again, which cells are the mutant ones really, and where do they originate from, and which lineage, to your point. So I do think this still needs to be done. We had tried to do this through FACS sorting, neurons versus glia. We were hindered, on my side at least, we were hindered because we work mostly with human brain samples, which are with very little control brain samples available to compare. So we've got to, our design is often not as robust as we would like. I think animal models might be better, frankly, to answer that question, obviously, for practical reasons. But yes, yeah.
Especially given that mTOR is a signaling pathway, that seems like it would be very, very important to find out where the originating cells are. Thank you.
Absolutely. Yeah, absolutely. I agree.
Jesse?
A patient with 3 mutations, was that inherited?
No, all de novos. So we tested the parents. Yeah. So, and I should have mentioned this. In all of these experiments that I showed, we test the parents as well, and they're negative.
So did you look, I guess it seems surprising that you had 3 mutations. Yeah. Did you look for any mutations like DNA repair?
That's a good question. No, because we are doing targeted testing, right? So we're not doing exome. We didn't do exomes on him because we only had fresh FFPE tissue. But it's a good point. Could there be evidence for that? Possibly. To date, so some of our kids, for practical reasons, some of the kids that we see, and both that get referred to us from out of state, oftentimes they'll come with a negative exome. So a lot of the time the providers want to, do some testing and they don't have brain tissue. So they'll do exomes, right? Or genomes now, maybe we'll see that. And it's oftentimes negative in the blood, at least. So at least we know if there's any DDR, mutation or something in one of these pathways. After now, so many have been tested. If there is some association, it would have popped up. But so far we haven't seen that reported. Yeah. Yeah, but it's a good question. We still don't know if it's these 3, how could these 3 mutations happen?
I just wanted to complement this. You probably didn't do phasing on the samples, right?
No, we did not.
That was my question.
Yeah,
we coming from the same parent.
Yeah. We did not do that. No, and also again, I think we had like lots of DNA concerns about like you completely exhausting our samples. So once we found these 3 mutations, we kind of like stopped at that point and just let it sit because we did not want to. We have very little DNA left, I think, on this child, but it's a very valid point as well. Are they on the same allele or not? Yeah, absolutely.
I was just wondering, with respect to the overgrowth aspect of this, do you think that your ability to find mosaicism is enhanced because A, you know where to look, and B, because it's overgrowth, you have cell proliferation, and so perhaps that could, right, just in terms of finding relevant mosaicism, do you think you have an easier time? Not that it's easy, but
yeah,
might other types of mosaicism be even harder?
I definitely believe mosaicism can be even harder than this. I agree with you because, I mean, the clinical features, what has helped is the clinical studies that preceded all this have really done a great job, as much as it was also, they described them in very different terms. But regardless of that issue, I mean, that's semantics in a way. They've done a really good job at describing these phenotypes. So we have a clinical eye for them. Like when we see these kids, they stand out. It's not questionable. what they have clinically. But the challenge has been is that the level, mosaicism came, even with these disorders, it comes with its own challenges. Because, for example, the brain is one thing, but actually with the skin overgrowth phenotype, so there's been a lot of studies that showed that in fibroblasts, oftentimes you don't find them. You have to test adipose tissue. It's actually the adipose tissue that's underneath that's mutant. And adipose cells are harder to work with than fibroblasts. They're not as robust in the lab to, isolate and grow and things like that. We had to do that with one case that was negative, and we couldn't find the mutation in the fibroblast. So we kept testing the fibroblasts for a while because they're just robust, right? They're stable. They're not going to go anywhere. And we couldn't find the mutation. So we felt like it must have been adipose tissue. And actually, Les Biesecker from the NIH has done similar studies in this overgrowth disorder called Proteus syndrome, which is caused by AKT1 mutations. And he has done once this very fascinating study of like whole body analysis of like multiple cell types, and it's all mosaic, right? And it's amazing how low it can be in some of these tissues. So as a clinician myself, not as a basic scientist, because I think the basic scientific aspects of this are truly fascinating, and I wish I could be talented to study those. But as a clinician, it comes back to me, like for me, from a practical perspective, is when I see a child, can we find the mutation to help the family, understand the diagnosis, remove ambiguity from their lives, and can we do it in what is the least invasive way possible, which often means a fibroblast, and that's often flawed. So I think there are definitely, there's a hidden part of this iceberg here for mosaicism that we're not tackling, I think, in the clinical setting. Yeah.
I had a question. It was really interesting with the sort of non-cell autonomous idea that there could be a sort of small set of mutant mosaic cells that cause the bigger phenotype. I don't exactly know how you would test this, but if you could cut out just the mosaic cells, do you think the rest of the cells that are abnormal, do you think that would sort of fix itself without the presence of the signaling, presumable signaling from the mutant ones? Or?
Yeah. So there's a mouse model that another group has with Proteus syndrome, actually, where they demonstrated non-cell autonomous effects in a mouse model. So we actually have and I didn't show this case. We actually had a very interesting case that suggests a test that maybe it's non-cell autonomous, which is a child who had two resections, basically. They had a big lesion with the highest mutation load. So it was like, I can't remember, it was like either 10% or something mutation level in that mutation. Resected, we looked at it, we looked at the EEG, marker from that region, and we looked at the even phospho-S6 biochemically, we looked at phospho-S6. We saw, mutation positive, EEG was abnormal, epileptiform, all that good stuff, phospho-S6 positive. The child after the surgery, so this region was removed. This child after surgery, shortly thereafter, had seizures again, right? So then went back in for a more extensive resection, and we did DNA diagnostics on the second surgical field, and it was like, less than 1%. But at the same time, it showed a lot of, it showed some of the cellular abnormalities. So it kind of gave us like a bit of a clinical glimpse, and the child was seizing, obviously, and everything. So it kind of gave us a clinical glimpse that maybe it is not cell autonomous, right? In this situation. So yeah, I think it's a definitely valid hypothesis, but we still have, we need more, I think, we need more data points from these kids. We need more, I think, like really either single cell, analyses to happen, or even if it's like at the macro dissection level, we need to like really kind of try to do more mutational profiling than what we've been able to do so far. I don't know if I answered your question, actually.
Yeah, I think so.
Yeah.
Is there any other question? Okay, I guess my last question is just like a speculation. You have an idea about what can be still explained with the 50% that from the unsolved case that can be studied. Yeah. But do you think there are still genes that need to be discovered as well, or do you think it's more like lack of methodology for the other alterations?
Yeah, so I was thinking for a long time that it's other genes, right? Like that we are missing other genes, other pathways. I am, with more time, I get more worried that we are missing, it's all mosaic in the same genes, and we're just not going deep enough, and we're never, I don't know if we'll ever go deep, at least in the humans, human setting, if we'll ever be able to go deep enough. So, but we've underutilized, I can speak for our group actually, but other groups I think might be different. We haven't still done, we started to do whole genome sequencing on brain samples. We've done deep whole genome sequencing. By deep, I mean like 90 to the 100x on a cohort of 15 kids who were unexplained, and we had brain and blood, and we didn't find anything, but that was short read. We haven't done long read sequencing. We haven't looked at structural variants. We haven't looked at, methylation abnormalities, which are possibly, present in some. So there's a few technologies we probably should throw at this problem that we haven't yet. But I've had a lot of hesitation from colleagues who utilize these methods, like even our long read sequencing folks and everything. They've had a lot of hesitation about mosaicism. It just makes them nervous. They're like, yeah, we're not sure we want to touch this yet or spend money on it. And so it's kind of an interesting, it's an interesting challenge. Yeah.
Okay. Thank you so much.
Of course. Thank you. Thank you, everyone.